He works on integrative modeling. He used to be a metadynamics person, and then
Right.
I think he became completely on uh, what shall I say, like integrative modeling person.
Yeah.
Okay. Okay.
I
So
have attended
today,
a few of them of those
uh,
seminars. I'm not sure whether today I'll be able to attend or not. I generally ask Anand to share the recording, and he never does.
No, actually, those things are online. Actually, they
Oh,
have put.
okay.
He has maintained the website very nicely, so those
Okay.
things are
So the
there. And
recordings are already there in the
I
website.
think he records everything, and I have at least got one meeting that
I see.
that I could not attend.
Then I'll remind him, or I'll find out. Yeah.
Yeah,
Okay, then maybe we start.
yeah.
yeah,
Yeah.
introduce.
Right. So yeah, it's good afternoon, everybody. Welcome to this CBMS Departmental Seminar, and it's pleasure to have with us Dr. Jagannath Mondal from TIFR Hyderabad. Of course, it would have been much more pleasure if he was here in person, but due to the situation, of course, we cannot have that. I welcome him to visit us sometime in future when things become much better. So just a quick introduction. Dr. Mondal did his master's in chemistry from IIT Kanpur in 2006. And for his PhD, he joined the group of Professor Arun Yetiraj in the Department of Chemistry in University of Wisconsin-Madison. He completed his PhD in 2011. And during this period, he has been working on coarse grain models and Monte Carlo and molecular dynamic simulation techniques for different kinds of amphiphilic macromolecules. And subsequently, during his postdoc, he joined the group of Professor Bruce Byrne. Of course, all of them are stalwarts in this field. So he was in Columbia University with Professor Bruce Byrne for about 4 years. And there he started working on, again, wide range of problems, free energy of protein drug binding, polymer osmolyte interactions, de-weighting transitions, and so on. And actually, I've been always impressed by the breadth of the topics covered by Jagannath. It's very impressive. And he joined TIFR Hyderabad in 2015, first as a reader, and currently he is an associate professor there since July. And as I said, his areas of interest is quite diverse, but broadly it is computer simulation based studies. He builds models, he builds coarse grain models and simplified models, chemically motivated models to look at a wide range of biochemical and biophysical problems. I'm very fond of his papers on Markov state models, where he looked at protein ligand interactions, molecular recognition, and ligand binding kinetics. And he has been also developing models at a genome scale, really large scale coarse grain model system. So it's very exciting, the kind of problems he has been looking at. He has about 60 publications to his credit. So now today, he's going to talk to us about interaction of short chain biomimetic peptides with membranes, lessons learned from computer simulations. So I welcome Dr. Jagannath Mondal to present his talk. Thank you.
Okay. I think it's a pretty elaborate introduction for me, and I'm pretty happy to share my research at SN Bose. And myself and Shuman has extensively met multiple times. We have discussed many of the directions, because we have many of the works which actually come in the similar domains. And as Shuman tried to point out that it would have been good to visit the center in person for a seminar, but I think I once visited actually when I was looking for jobs and all these things. But but basically it will be a pleasure. Maybe in future definitely I'll stop by if I could. And but the point that I'm trying to make is that as Shuman tried to point out, I try to use whatever I learned from my my PhD and postdoc experience, and try to see whether we can try to find motivations to do interesting directions, which is basically comes under the domain of chemistry and biophysics. And we try to use computer and try to use some of the developed theoretical development, which is quite you can say archaic, but they are pretty what shall I say, relevant even now. And whether we can try to see whether we can try to encourage ourselves to learn all these things from there and see whether we can try to understand the most integral part of our life, which is basically many of the biophysical related problem. But what I think, as Suman tried to point out, if you can see my cursor, so this particular what I have done here is that I have tried to show the kind of projects that we try to pursue in our group, everything by computer simulation. Some of them basically have some experimental collaborations that is happening more of late. And as Suman tried to point out, so one of the topic which I basically pursued extensively and basically I like most is this interaction of protein with ligand binding. But I thought that Suman has heard multiple times speaking about that from my side, and maybe it will be better if I look into other things for this seminar, especially the topic that we are going to talk about today. We are going to look at whether we can understand the interaction of membrane with small peptides and try to see whether we can see whether how computer simulation can play an important role and whether we can learn something out of these things. So that is basically what we are going to explore today. But as Suman pointed that we also try to think about whether we can try to even look for even a larger scale system like genome like bacteria and all these things, something that I learned from my PhD advisor Arun Ethiraj, to think everything from a perspective of polymer physics and see whether we can develop models which are relatively simplified but still effectively can bring out the essential physics underlying these things. So I'm not going to talk about none of this. So we are going to focus mostly on this area of peptide membrane interactions. But the point is not only this. Point is that how we can actually try to use computer simulations, which and try to understand something which might not be so trivial at its at appearance. So what I think we are trying to do in our group, we are trying to see whether we can look at chemistry from the perspective of different types of models. It can be atomistically detailed. It can also be coarse grained. By coarse grained I means that you can think about many of the chemical atoms can be approximated effectively to a particular mean field approach, which are basically called this big spheres, which you can super atoms. You can think about coarse grained models, and then whether you can even think about for the problem that you are interested in, whether you can further approximate the model to make it even more simplified to a mesoscopic model, and see whether you can numerically simulate the behavior of these kind of systems using one of the popular techniques, which is called molecular dynamic simulations. And you can also try to learn many of the observations and try to see whether you can understand them from the idea of statistical mechanics or statistical model building like Markov state models, and try to see whether you can get idea from thermodynamics and kinetics from those things. So in this particular aspect, what we are going to do today, we are going to look at one of the popular systems in biology, which is called antimicrobial peptides. So basically the antimicrobial peptides are basically an promising alternative to regular antibiotics that we generally use as medicines. It's mostly because antibiotics are becoming mostly, what shall I say, like resistance, and this is because the bacteria also gaining resistance. So people are trying to see whether you can try to use some other tricks to deceive the bacteria or any other potential harmful species. And one of the strategy in the community is that try to use peptides which are naturally created, and this can basically to some extent kill the bacteria or any other microbes, and see whether that can be used as a potential drug candidate. So as you see that this antimicrobial peptides are becoming quite popular as as we go on the time, and this is up till then, and many of them are under clinical trial. But there are also other aspect of these things. Many of these antimicrobial peptides actually also, instead of killing bacteria, it end up killing your red blood your red blood corpuscles also. That is not good. So that issue is always there, but still this is a potential alternative from the antibiotic perspective. So but what actually people have tried to look at, people have tried to see that whether you can look at naturally occurring, that means antimicrobial peptide, that means that peptides which you can get from the extract of the any other eukaryotes, and you can whether see whether those things basically has a activity to kill the cells or bacteria and all these things. And few of them are basically shown here. So these are different names, you don't have to remember any of these names, but the point that we are trying to make here is that many of these naturally occurring antimicrobial peptides are actually right now in clinical trial, but none of them has actually passed the clinical trial. This is mostly because, as I told you, that this actually has lack of specificity, and second is that it is sometimes it can actually get completely degraded inside the cells, then it cannot do the function. So one way that chemists who are actually now bioorganic chemists try to see whether there is a way you can design synthetic peptidomimetics. So that is basically one particular strategy that people are trying to see whether one can design better peptides by synthetically modifying the regular motifs of alpha amino acids, and see whether one can design synthetic peptides which can do the job instead of a regular naturally occurring antimicrobial peptides. So that is one of the thing that I started learning. As someone pointed out that I was trying to do my PhD in University of Wisconsin Madison. So one of the way I used to get my funding is because I used to be part of a center called Nanoscale Science and Engineering Center, which was led by one of the bioorganic chemists. His basically name is Samuel Gelman. He actually is popular for thinking about one of the particular ways of modifying normal alpha peptides or alpha amino acids. So let's say you have a alpha alanine. This is left hand side I'm showing. What one can do is that in the backbone you have one carbon atom. So what one can do, this can be synthesized actually in a lab, and that is what Sam generally do. You can actually add an extra carbon atom on the backbone and try to see whether you can actually gain any particular advantage out of this. So one of the key advantages that Sam's group has shown from in the years of 1999 to 2000 onwards, that you can actually create many interesting helices which are short chain and they are still stable. And very interestingly, what one can do is that if you try to think from the chemistry perspective, you can actually have two carbon atom in the backbone. So you can even synthesize the cyclic amino acid which does not have any alpha amino acid counterpart. So these are called, let us say, ACHC, which is basically cyclic amino acids, but this is completely beta amino acids. And essentially, you can also have all kinds of beta counterpart of alpha amino acids. So essentially, this actually gives you an option to create even a rigid helical backbone with only few residues, and that has been one of the USP of their group's research, that they can synthesize very short rigid peptides which are synthetic in nature, but basically they can actually do the job of many of the other activities. And one of the activities that they looked at in their past research is that can these peptides, which are basically completely made by this beta peptide or beta amino acids, can we see that these are having any antimicrobial peptide or not. So what I'm showing today, this will be basically the center of our today's seminar. So what we are going to talk about is that we are going to talk about this short 10 residue beta peptides. So this particular peptide, basically you can see that there is one tyrosine. So this is shown by the cyclic in those red residue, so blue residues. And then you have this ACHC group, which are basically almost like cyclohexyl group. And that is these are here shown by these red colored residues. And then you have this side chain, which is this lysine. So these are basically just green colored residues, and you can see that this is basically the stable alpha helical fold, not alpha helical fold, they call it 14 helical fold of a particular peptide. So essentially the point that we are trying to make here, these are very small peptides, a small size peptide. If you look at the length from one to end, you will see that the length is about 1.4 nanometer. But the key thing that they try to do, they try to see whether if they inject these particular peptides against any other bacteria, so they tried certain gram negative and gram positive bacteria, what they try to measure is that how much of the concentration you need to kill this kind of bacteria. So essentially the numbers that they generally turn out from their experiment are basically a macroscopic quantity called MIC. There will be minimum inhibitory concentrations. So they basically show that in all these cases, this kind of peptides can kill many bacterial cells. One is you can call about E. coli, Bacillus subtilis, and there is also other bacteria. In all these cases, the concentration that you need is significantly smaller than a natural pretty prototypical alpha amino acid oriented antimicrobial peptides. So here is a better statistic. So what I'm trying to show here, the 7 and 8, 7 is basically the peptide of our interest. So this is basically the same peptide that I showed you in a, written in a more, chemically structured model. So this is 7, and you can see that against different kinds of bacteria, the activity that basically shows that within 12.5 milligram, you can actually kill a particular, particular concentration of bacteria. But similarly, if you look at the typical other bacteria, like let us say melittin, you need, pretty strong, either concentration or almost similar order of magnitude. So essentially it tells you that melittin, which is a pretty large, almost 27 residue, or 30 residue, antimicrobial peptides, is, basically having almost equivalent or even, high, lower activity than a very small chain peptide, which is basically called this beta peptide. So basically what they try to point out is that this beta peptide can be very strong candidate for a antimicrobial peptide activity. But what essentially they showed, that since they are chemists, they started playing with the sequences of this thing. So one thing can do is that one can simply scramble these particular chains, so you can actually lose, the entire sequence in a different way. You can rewrite it, let us say, as in the number 8, and you can show that if you try to now scramble it in a different way, this antimicrobial activity, which is measured by MIC, goes up, goes down significantly, because MIC becomes almost more than 200. So it tells you that if you basically try to play with the sequence, you can actually see that there is a significant sequence selectivity against the bacteria that you can show. So in all these cases, you can clearly see that compared to, the 8, the 7 is much better antimicrobial agent. So that is basically what they can control from their end, and that is what I think, one of the, selling point of Gelman's most of the work, that you can try to design sequence dependent antibacterial activity of this kind of beta peptides. So to, learn more those things, what they try to do, they try to even see whether these peptides are actually effective against fungus or not. So what they try to look at, so in these cases, they are looking at many such peptides. One of them is basically, again, 7 and 8 is again appearing. So 7 is this, this circled one, and this 8 is this scrambled one. You can clearly see that if you plot this, this is almost like measure of how much concentration you need to kill a bacteria. You can clearly see that, the scrambled one will take much higher concentration, and you can kill, much at a much less concentration this particular bacteria. So this is experiment actually what you generally get. So it tells you that these are having, as far as the minimum inhibitory concentration for killing bacteria is concerned, these peptides are, a bona fide antimicrobial agent, while the 8 is not. So basically that is where the job is done. It tells you that chemistry can control how you want to make the antimicrobial activity. But what we really wanted to ask ourselves, what we basically started, getting intrigued, and that actually started when I was trying to, work on my PhD in my Wisconsin Madison. So that question I had that time, and the question that I actually had is that how does this short chain peptides, which is so shorter, how that can actually still be effective, even better than these naturally existing antimicrobial peptides, which are definitely larger molecular weight or even longer chain. And the broad question is that I had is that only thing that I have from the experimental group is that they know the experimental MIC. Now the question is that MIC is something which is particularly macroscopic readout. The question is that is there a chance that one can actually think about, can we try to learn beyond this experimental MIC? The specific question that we try to ask is that how does this peptide actually actually act? So by action we mean that how does this peptide actually kill the bacteria, but before killing the bacteria it has to do something, which is it will interact with the membrane of that particular bacteria. So what we try to understand is that how does this short chain beta peptides can impart disruption to a particular biological membrane. So that is the question that we started thinking, and the question basically started appearing because there are multiple hypotheses which is present in the literature on how a antimicrobial peptide can kill bacteria. And this is kind of a slide that generally you can see whenever you look at in any of the in any of the literatures which talks about antimicrobial peptides, and there are many different hypotheses that people generally try to talk about. So in all the hypotheses, central is that these peptides, which are basically present, let us say, in solution, they have to interact with a particular membrane, which is the first line of defense for any particular bacteria, and then see whether it can actually undergoes any particular conformational change, and then most importantly, can it form some sort of can it go inside the membrane, can it form this kind kind of pore like structure, and essentially can kill this bacteria. So there are many different proposal, and one can think about all these sorts of different toroidal pore and barrel step pore, all these things. We are not going to the details. You can also think about detergent like actions. This peptide can actually be amphiphilic in nature, and that can also completely make sure that it can disintegrate the membrane. But the point that we're trying to make here is that this is basically a cartoon that people generally show, and these are mostly centric to a particular long chain natural peptides. Now the question that we are trying to ask ourselves is that we have in our hand at a very short chain peptide. The question is that how does this short chain peptide even encompass this particular entire bilayer that is supposed to be the membrane's essential building block. So that is the question that always intrigued us, and that we thought that why don't we try to understand. So again there are different cartoon that you will see again in literature. All these things, these ports are more nicely designed by different experiments. This is how how they look at. It's very difficult that you will find that many experiments. We'll get into the details and try to show you what kind of pore is basically being formed, and whether there is at all any pore formation or not, that is also another question. So what we try to look at when we started looking at this thing, and I'm looking at from a almost like you can say that this particular problem I'm trying to look at from a while, and you will realize soon actually. So what we started thinking is that one particular way one can think about, maybe we can try to do computer simulations. We'll try to do a long time simulation of how these small peptides, let us say at a very dilute concentrations, actually interacts with a biological membrane, and try to see whether we can learn something out of these things or not. So one normal strategy that people generally take is that we cannot model a biological membrane. So what one can do is that one can try to use a mixed phospholipid bilayer, which is generally considered that in your biological membrane of a bacteria you will have sort of this kind of phospholipids, which is one of the two of them basically which is popular, which are called POP and POPG. I am not going to go through all these details. There are debates which is a good model for bacterial membrane, but generally this is what people generally use as a very minimal model for bacteria. And the question that they try to ask is that how these peptides are going to interact, and at a higher concentration what will happen. And typically these kind of simulations are pretty straightforward, and that's what we try to do at the very beginning. We actually try to use regular charm 36 force field for lipid. In my post PhD group, as I was telling you, that we were trying to do collaboration with Sam Gelman's group. We even optimized the parameters for beta peptide. That was one of my group members PhD thesis, and we could basically now stick to that similar kind of force field, and we can try to look at how does this single peptide actually interact with this particular membrane. So this is a problem that we thought that we'll first look at, and maybe if we wait and see, maybe we'll learn something out of this thing. So that was the point that we started doing the simulations almost 3 to 4 years before. And one of the things that we'll see in this entire lecture is that how do you vary the concentration of peptide. So we'll basically use this particular quantity called P by L. This is the peptide to lipid ratio. As you increase more and more peptide, this particular ratio will also modify accordingly. And so basically what one can do, I'm not going to show you in particular movie. What one can do is that one can actually use one particular peptide, and you have this membrane surface. I'm not going to show you a movie. If you do this simulation for a microsecond long, it definitely used to take more time, at least 3 to 4 years. Now with GPUs it will take even lesser time. So essentially you will see that this particular peptides, since this is amphiphilic in nature, it will basically immediately come, and basically within a microsecond or even less than microsecond, you will see reproducibly these particular peptides are going to get partitioned near the interface. And that's where it basically stops. If you extend the simulation, nothing will happen. You will see that these hydrophobic groups, which are the cyclic amino acid groups, are going to face the lipid tails, the lipid center. And then this is the hydrophilic group, which is the green colored lysine groups. You will see that they are going to face the phosphate and the water interface. So this is typically how the amphiphilic peptide is supposed to interact, and you will see nothing new out of these things. But this is good that at least you will say that your force fields that you basically put in here most likely is doing the job that is supposed to do. It's like oil water partitioning. It is just getting partitioned in this particular interface. So we thought that, okay, we learned something out of with some single peptide chain. Why don't we try to put multiple of this particular copy? Now the moment you start putting multiple of this copy, one is technical problem that we will start seeing is that because of the periodic boundary conditions, you have to use even longer and longer water layer. You can still stick to a smaller bilayer, which is another concern. But you have to stick the, you have to basically use a very longer water layer, which will increase the total amount of number of particles that you are going to do in your simulate in your systems. And as we do MD simulations, we know that total number of particles is always a concern for our case. So what we tried with this large system, we thought that we'll try to run it for longer time. We tried to run for 2 microsecond long, and what we basically found that it almost takes more than a month. And even with that, we find that this is basically where it ends. It just gives rise to, at a very high concentration, a sort of an aggregate on the top of the surface, and that's where it ends. Some of them get interfaced, partition inside this, and you will see that, but on top of it you will see a very big mess of these peptides which are aggregating. And this is nature of this kind of peptides. It will aggregate because you have hydrophobic surfaces. But this is where this simulation basically comes to a end. And even if you extend it, let us say if you extend it for 3 to 4 microseconds, which you could do at that particular point, nothing happens. It just sticks there actually, and that's pretty much where you just end. So the question at this particular we had is that, okay, shall we, maybe we can abandon this project here because we are not seeing any pore formations. Maybe our force fields are fine, but maybe our computer time is not good enough. Or we can see whether we can try to make a trade-off, and can we try to learn from this particular situation of computer simulation with all atom models, which I generally always go for. But for this project we found that it's not doing much. It's basically just stopping it here, and we can extend it, but then that way I don't know how long I have to run. So basically we thought that why don't we look at what actually is going on at up to 2 microsecond layer. We found that you can plot one particular way one can check is that one can plot something called water density profile. And what we see is that water density profile wise, in a neat membrane, and in a peptide presence membrane, there is not much differences. There is a very small, as you see in this by this big sphere color thing, there is a small intrusion of water. There are partial water portion that is present, but no peptides actually get inside, and at least does not happen during the simulation period that we are running actually. So then we thought that if you are really looking for a solution, can we think about, can we make a trade-off? Can we try to learn from our lesson, from this particular situation of all atom simulations, and can we look for a computationally cheaper solutions? So one of the way people think about is that we can actually reduce the resolution of our model, but effectively still try to maintain the chemistry a bit. And one of the way to do it is that you can try to develop something called coarse grain model. So that is basically the question we started asking ourselves. And as we try to point out that what actually happens, so if you look at a very simple example, ligand binding and unbinding in a particular process, one can see that one can basically, this kind of process definitely takes quite a long in time. It's mostly because the surface that we look for is pretty rugged in nature, the underlying potential phase space. So one way that you can overcome these kind of things, maybe you can try to smooth this out. And one of the way to smoothen the surface is that you try to develop models which are simplified in nature, so that overall underlying potential surface is automatically smoothed out. So that is one thing that one can think about. So this is exactly how most of the coarse grain model try to are motivated. And one of the popular coarse grain model which is out there, which is called Martini coarse grain model. So in this Martini coarse grain model, which is basically developed by this group of Marink and co-workers, so they are doing it from almost 2007 2008, and these particular force fields are quite popular to model systems which is basically for protein, lipids, and all these things. And these are there for a long time, and we thought that why don't we try to see whether we can try to learn from these particular systems. At least this will be computationally cheaper, and maybe we can run it for longer, and can we see anything we learn. But and most advantageously, like the way Martini works is that instead of having, let us say, these long chain peptides or anything, you basically reduce the resolution by mapping 4 atoms into a big one coarse grain atom. So let us say this is my coarse grain atoms, and similarly for peptides or alkane you can do the same thing. And this is almost like you are trying to build a Lego, and you are trying to learn from whether this can still effectively give you the essentially similar kind of molecule, but reducing the complexity of the real molecule. So that is basically the architecture. So it does not look really nice like what you generally see from a in atomistic representations, but basically you can still see that this kind of ball and stick models are to some extent there. So it gives you some idea of how the secondary structure and tertiary structure looks like. And what actually has Martini has already done, it has efficiently mapped for most of the alpha amino acids very extensively. All the lipids are also very well mapped. And what they do is that they just don't only map it, they try to also parameterize their non-bonding interactions against experimental, let us say, oil water partition coefficient for similar residues, and try to see whether you can actually map these things from the perspective of a regular, well-known interaction potential like Lennard-Jones electrostatics bond angle and dihedral. So this is exactly what we thought that may be something that we can go about. And what we did is that we thought that why don't we think about coarse grain mapping this beta peptide. The only thing that I try to tell you that beta peptide is not exactly like an alpha amino acids. So this is slightly different. So we have to really think about a different mapping. So we had to map these things out ourselves. So what we did is that we know that there is an extra carbon atom in the backbone. So we used to use, instead of 4:1 mapping for the backbone, we used a 5:1 mapping. And then backbone atoms and the corresponding dihedral will be different. So we had to do all atom simulations for single beta peptides and try to map it to a coarse grain molecule. But fortunately, the side chains does not have to be changed. It's mostly because, as I told you, the side chains of the beta peptides are same as alpha corresponding alpha peptide. So that way you don't have to, you can just directly borrow the parameters from Martini force field. So similarly, lysines and tyrosines, you can easily take it. Now ACHC, which is cyclic amino acids, it has a it has a side chain which is similar to cyclohexane, and for cyclohexane, Martini already has parameterized things. So what one can do, if you really now look at your corresponding Martini representation of my beta peptide, this looks like this actually. So this is does not have all the atoms, but it does not look so bad also. It maintains, after the optimizations, it maintains its rigid helical nature. And the advantage is that you can also try to use water models, which is also present in explicitly in Martini. They are again not so atomistic. 4 water model is mapped into one, and they have also something called polarized water model. I will come to this particular point later. And in principle you can use those things, but the advantage that one can do is that now you can actually have much less number of atoms in your system, and you can actually simulate it for a long time and in a larger scale. So this is exactly what we try to end out. But the first thing that we have to see whether how do we validate our coarse grain model against an atomistic model. So what we did is that we first looked at the same thing. Can my coarse grain model mimic the basic thing, which is a single alpha amino acid, sorry, atomistic model does? So one can look at whether if I start from a bulk phase, can coarse grain model also show this kind of adsorption to the interface? And we see that it basically, this is the last eventual snapshot of a single beta peptide getting into the membrane, and we can clearly see that these two looks pretty similar. This is my atomistic model. We have relatively smaller bilayer slab. This is a coarse grain model where we can actually do it for even larger bilayer slab, and you can see that this particular coarse grain peptide, also similar to this one, also gets partitioned near the interface. Again, you can see the red part, which is the cyclic groups, are actually facing the the hydrophobic lipid tails, and this green part, which is the hydrophilic part, they are facing the phosphate group, which is the charged residues. And essentially this is matching because qualitatively it tells you that this hydrophobic hydrophilic match is at least there in coarse grain mapping. You can even try to look at one of those trajectory where the where the peptide is coming towards the interface, and you can see that the trajectory does not look so bad, but you can clearly see that in a coarse grain model these things are a bit more smoothened. As I was trying to point out, in atomistic model, the trajectory of approach to the interface is more or less more rugged. Now what one can do is that this is a very qualitative agreement, but what one can do, one can actually look for a more quantitative agreement. So what we try to do is that we try to look at how much free energy it gains in an atomistic model versus a coarse grain model for adsorption to the interface. So this is exactly we thought that might be a better match. So what we did is that we carried out many simulations in which this kind of process was simulated, both in atomistic approach and in coarse grain approach. And then what we did is that we tried to develop a Markov state models where you can figure out in a two-state model what is the relative population of this solvated state versus this adsorbed state. So if you know the population which is shown by this pi in both the state, what is solvated by the adsorbed state in solvated state, you can then calculate the delta G of adsorption, which is basically the ratio of minus kBT ln of this particular ratio, which is pretty straightforward. So in Markov state model, we try to do a very simple two-state model. We try to calculate something called equilibrium or stationary population of these two state, which is solvated state and this adsorbed state, and we try to see what is the delta G we get. What we found is that atomistic models gives me a delta G of minus 9.3 kcal per mole, while the coarse grain model gives me a delta G of adsorption which is minus 8.97. And I was pretty happy to see this because I did not even expect that this will be so close. But it tells you that even in a semi-quantitative approach, this particular models does the job it is supposed to. It can at least show that quantitatively the atomistic adsorption and coarse grain adsorptions are giving you similar kind of scenario. So then actually you have some what shall I say, benefit of doubt for this coarse grain model, and you can go ahead and then try to look at a higher concentration of beta peptides what it does to the membrane. So this is exactly where we, if you recall, this is exactly where we stopped our atomistic model because we could not run. But now we have similar system but in a more coarse grain representation. So we have less number of residues, we have smoother surfaces, so we can run it for now longer time. So this is exactly what I'm trying to show. So in this case we had, let us say, 5 peptides. Initially all the peptides were in solutions, and then we put it in even a larger bilayer, which is 336 lipid bilayer. In all atom we could go maximum 128, and then we could run it even for 20 microsecond in coarse grain time scale. So if we try to now show you a particular movie what it does, so this is basically what we see. Something went wrong. Okay, I think my PowerPoint went off. Give me a second.
Yeah, you can just restart the PowerPoint.
Yeah, just see actually. Yeah, so yeah.
Maybe the movie was too heavy for it.
So I'll just show it. I'll just can you see the presentation again?
Yeah.
Okay. Let's see whether I can play it now. Actually, not. Yeah. So I can now see it. So particularly what I'm seeing, I have this brown color thing. These are some waters. The big spheres are phosphates. And initially I'm seeing it for the first 1 or 2 microseconds. The peptides has already gone towards the interface. And then if you just go on for this particular simulations, what you will start seeing is that eventually some of them basically aggregate together, these peptides. Now it is aggregating in the interface, unlike the what you saw. And after some time you start seeing that they are almost becoming vertical. And then it is those big spheres which are water molecules. You start seeing that those waters are actually kind of permeating across the membrane, and all the peptides are basically to some extent encompassing both the bilayers. So that is something that basically you generally try to say that this is something that it is basically getting inside, unlike what you saw in all atom in a single 2 microsecond simulation you could not see. But in a coarse grain representations, what we could see is that in a longer time scale you can actually see this kind of peptide getting internalized and basically making very disordered port, not very stable port, but basically it's making a disordered port at a pretty what shall I say, very low concentration of peptide, which is 5 peptide at 336 lipid, which is much lower than what we initially tried in an all atom representations. So if you can let it go, you will see even some of the phosphates which are also getting disrupted, and they are also getting inside the membrane actually. So so what we did is that it actually gave us a clue that maybe with a coarse grain model you can now vary the peptide to lipid ratio. That means you can increase the peptide concentration on the similar kind of bilayer and start seeing what actually can do. So exactly this is exactly what we did. So now if I go to the next slide. So what we try to look at, we try to first look at what is actually happening to these, these peptides. So one way that you can actually characterize this thing is that you can try to calculate the distance of each of the peptides. You have 5 peptides towards, let us say, the lower leaflet of the peptides. And then you can also calculate something called the tilt angle of the peptide with the membrane. So let us say this is your normal and this is the tilt angle. So if you try to follow the total time scale, so we ran it for almost 40 microseconds to see whether this remains stable or not. So we found that at least 3 out of 5 peptides, at least 3 peptides are basically very easily we could see that this peptide initially was, let us say, in the solvent. After a particular transition at 5 microsecond, we started seeing that they actually get inside and basically this remains stable there. More interestingly, what we found is that if you look at the tilt angle, so if you are at the very beginning, you will be at 90-degree angle. That means that you are actually almost in the normal, which is basically flat to the surface. And after 5 microseconds, when it peptide got inside, you will see that they are actually changing their orientations, and it's becoming almost like 30-degree, which is almost like perpendicular to the surface. And this is obvious for both of them. So two different things are happening, because this is the vector that we are basically plotting. So you can actually see that in one of the cases it will go to 150, in one case with 30. But essentially this is the same thing that is telling you. It is saying that it is changing the tilt angle, and that is very interesting. It tells you that when you basically have them in completely parallel orientations, they are not really what shall I say, in a way to make the pore. But essentially, once you have many of them, they actually start changing their angles, and then you start seeing that they are actually making in vertical orientations, and then it is actually bringing those waters, and that is basically remaining there. So that actually kind of gave us a clue that maybe we can now increase the concentration of the peptide and try to see what actually is happening. So these are two independent trajectories that we now did with a longer higher peptide to lipid concentrations. And what we could see is that in both the cases, within 20 microsecond simulations, you will see this kind of pore that is basically there, where you have lots of water which is present, and then you have this kind of pores which is formed. Even what you will see is that at a higher concentration, you will see two pores actually being formed. And essentially, if you wait for a long time, you will see that those pores are actually getting what's smeared into a single pore actually. And that is basically tells you that there are two things what is happening. One is basically the time scale that you can actually access in these particular things is relatively much longer than atomistic representations. And more important, the size of the membrane also matters, essentially, where you can make sure that you can actually increase the larger lipid patch, and then you can actually go on for a longer time. So this tells you that with higher concentration, the peptide of the peptide, you can see this peptide pore formations even much even in a what shall I say, more reproducibly, and you will see that they will happen even much lower much faster rate actually. So what we can do is that we can actually now go on and try to individually look at what is going on with these two different concentrations. So yeah, so basically we can then start varying this the similar kind of distance and the tilt angle criteria. So this is now, if you compare for all the different concentration of the peptide to lipid, and you can also look at the distance, minimum distance, you will see that at a very low concentration, where you have only one peptide at a particular 388, which is the very large peptide to lipid, very low peptide to lipid ratio, peptide remains on the top of the surface, it does not get in. But if you start increasing the concentration, you start seeing that this peptide, all the peptides start getting inside. And if you increase it further, you will start seeing that this time scale even gets even faster. Initially, it was, let us say, at 5 microsecond. On effectively, what you will see is that this is happening even faster. And as you see here, that in some of the cases, some of the peptides come later, and in for 1:22 ratio, you will see that this particular pore formation actually finally gets coalesced into single pore. The same thing you will see for the angle. You will see that at the very beginning you are at 90 degree. That means that peptides are flat on the surface. Once you increase the concentration, you will start seeing that the peptides changes their orientations very quickly inside the membrane. And as you increase the concentration to peptide to lipid ratio to even higher, you'll see that this change in the angle happens even a more, what shall I say, even a more faster rate. So you will see that in those things are almost now within few microseconds, which is also correlated with the distance inside the membrane. And you will, within few microseconds, you will see that they are changing the angles, almost becoming verticals, and that way it can encompass the membrane very nicely. So this actually also can be quantified. You can do one thing, you can calculate the density profile of the membrane, of a density profile of the peptides in the phosphate. So at a very, when you are at a very low concentration, when you have single peptide, which is shown here, you will clearly see that this particular density profile tells you that this is just encompassing the phosphate bilayers. As you increase the concentrations, you will start seeing that peptide to lipid ratio, the inside the two phosphate, it is encompassing both of them. That means that it is there, it is basically spanning both the leaflets. And then as you increase the concentration for peptide, you'll see that this density inside the membrane actually also increases significantly. So this tells you that peptide to lipid ratio is acting like a cooperative process, where you start seeing that at a very low concentration nothing happens. All on a sudden you start seeing that at a threshold concentration, which in our case is 1:67, peptides start encompassing the leaflets like a transmembrane probe. So this is basically very interesting. Why it is interesting? Because, and this is basically the summary of all of this. You will see that at different concentrations, as you start going from 0 nanosecond to 20 microseconds, you will see the progression of pore and waters, which is basically present. So what is actually interesting is that, in all these cases, there are lots of water which is coming inside. So what you can do is that you can calculate how many waters you are basically seeing, at least up to the half of this particular, let us say center of the lipid and center of the lipid bilayers, and try to see whether there is any, whether you can calculate this kind of thing. So what one can do is that one can try to, for last two concentration I showed you, which is the higher concentration, you'll see that initially there are very less number of waters inside the center. Once the pore formation is completed, you will see there is a sudden transition. You'll start seeing that there are lots of water which is going up to the middle, and even in 1:22, it is pretty obvious that with increasing time, this number of waters getting inserted is pretty large. So this is something that experiment generally look for. This is called water leakage inside the membrane, so that peptides comes, it basically creates defects inside the membrane, and then it makes sure that water gets leaked, and that way it can solubilize the membrane. And this is exactly what we also started seeing, that these peptides, even though they are short in size, still they can basically get inside, and then they can actually ensure that water is actually inside. So there is one particular interesting thing that one can see. This is called rare event, that you can see that in some of the cases, some of the water makes a full pass. You start from, let us say, one leaflet, and then you try to count whether it has a chance that same water, if you track it, can it go past the other leaflet. And we show that there are multiple events that is happening. So at two different concentration we looked at, in both the concentration, we saw that there are at least pretty significant number of this kind of situation, which is called membrane permeations, where you can clearly see that all these cases, the peptides are getting sort of many of the waters are actually starting from one leaflet and getting outside. And this is one of two of these representative trajectory of two water molecules. You can see that, let us say you start from lower leaflet, which is this blue color thing, and essentially it basically make sure that it finally gets into the other leaflet. On the other hand, this is another trajectory. It starts from, let us say, the top leaflet, and essentially it basically makes sure that this water gets into this thing. And what we try to look at is that how long it takes for a particular water molecule to get from, let us say, one leaflet to another, and that you can measure. What we found is that essentially the time that it takes is within 10 nanoseconds, but you will see very long tail also. There are many events which is taking even 50 to even in one case we saw that it also takes almost 250 nanoseconds if you really track it before it can pass through the entire membrane. So this is also very sluggish, and that is essentially true because you are almost going through a hydrophobic environment, and that is going to take more time. But essentially what it is telling you is that it is pretty difficult to do it in a completely peptide less bilayer. But once you have a peptide which can make pore, it can ensure that, just like ion channel, it can transport the ion water from one side to the another side. Now the question is that how do you basically characterize this membrane deformation? One way to that you can look at is that how the phosphate density profile looks like. So when you have a neat membrane, which is this 0 to 388, you'll see that in both the cases the membrane density profile of the phosphates are very sharp. As you increase the concentration, which is expected, you'll see that this density profile starts getting smeared. That basically means that many of the phosphates also start getting inside the membrane, and they are basically almost getting smeared, and that is pretty obvious. And many water molecules also, this is the water density profile. This is again reflective of the previous result, that many water molecules also get inside these particular things. So this is actually also reflected in the membrane disruption. This is exactly what generally people look for. We look for, let us say, how because once the peptides gets inside, you can see that how it is affecting the membrane bilayer. So one way to measure this thing, you can measure the something called bilayer thickness profile. So if you are in a uniform lipid where you would have no peptide, you will see that the bilayer thickness is pretty uniform. In all the places you will see that the bilayer thickness is same. That's why this is shown there is no particular spot. The moment you start increasing the concentration, you start seeing that there are certain change in the heterogeneity in the thickness. And as you increase the concentration significantly, you start seeing that many of the locations basically are completely punctured because the peptides are getting inside and they are creating traps, and that is why you have pretty low insertion depth or the bilayer thickness in those locations. Another way that you can look at is that you can look at lipid order parameters. There also you will see the same thing. The lipid orders are getting more and more disordered. That basically tells that these peptides actually also are not only changing the thickness of the bilayers. It is also making sure that the lipid orders are not present, and that basically means that overall it is doing a job what an antimicrobial peptide is supposed to do. It is essentially trying to make sure that the disintegrity of the bilayer is pretty much completely disrupted actually. Now here there is an interesting question. This is exactly what I think this coarse grain model actually teaches you. So initially I am telling you only the success story of these things, but essentially we started working on these things on Martini's regular water model. So as I told you that Martini is a coarse grain model, but unlike many other coarse grain model, it considers water also explicitly. But the way regular Martini water works is that it mimics four big, let us say, TIF3P water molecule into a uncharged water model. So this is the regular Martini water model. So initially we tried to do a simulations with this particular Martini uncharged water model, and this is the more upgraded version, which is basically called Martini polarizable water model. So in this particular water model, they try to make sure that the electrostatic part is also incorporated even in this 4:1 mapping, and this is exactly what we have used in current work. But if you try to do the same simulation with this uncharged water model, you will see that even if you increase the concentration, you will see that the peptide does not go past the membrane. It basically just gets stuck in these particular cases if you basically use the regular Martini water model. So it tells you that the electrostatics of the water is actually key for this kind of porations to happen. And this also tells you that if you really want to take a coarse grain model with some belief, one need to really test and try properly. And the electrostatics is one of the key points of Martini water model that one need to really investigate, and that actually can also give you completely different results. So that is also something that we learned on this process. Essentially we did all the simulations at the very beginning with this uncharged water model. Nothing happened, and then when we started optimizing our models with this this polarizable water model of Martini, we started seeing that this pore formation becomes actually evident for all the simulations. So that is one advantage. Now come to the key question. That is the question that we started thinking about. As I told you that these peptides are very short and they are not supposed to encompass the entire bilayer, then what is actually happening that is ensuring that this peptide can still completely become perpendicular and still basically not tilted. It is pretty much trying to encompass this membrane. So what we found is that from our simulation we try to measure this end-to-end distance during our simulations. So if you do a simulations of this peptide in pure water, you will see that the end-to-end distance, as I tried to point out at the very beginning, it is fluctuating about 1.5 to 1.6 nanometer. If you start thinking about peptide and lipid at 1:388, where you have only one peptide on the membrane, you will start seeing that the peptides end-to-end distance does not change. But the moment you start seeing this pore, what we found is there is a correlation. The peptides get extended actually. So initially it was at 1.5. You will start seeing that this peptides becomes extended up to 2.5 nanometer, and that is actually very interesting. So it tells you that even though in a normal water these peptides are very rigid, but if you put it inside the membrane, this peptides try to stretch it out so that it can reach out to the other triplet, and that will basically be the case because you have charged residue. It is going to reach out to the phosphates, which are basically also charged, and that is exactly the motivation for these peptides to encompass this particular bilayer. So you can also now look at the distribution of this end-to-end distance. This is only in water. As you increase the concentration, you will start seeing that this end-to-end distance distribution gets shifted towards a higher value, and what you will see is that these particular peptides are now getting stressed. More than that, you have lots of water which is acting like a pad, and it is making sure that it can anchor this peptide inside this membrane and can hold it out, and then it can maintain this vertical representation, which was supposed to be crucial for this kind of membrane to see any pore actually. So this is actually one particular observation that we made in this thing, that interfacial water layer is acting as an anchor, and these peptides are getting stretched, which is ensuring that this peptide pore formation actually possible, even though these are much shorter than a natural peptides. Now the question that we actually had at this particular moment, okay, we are seeing all these interesting results. Are these results are coming for these are all right results for the wrong reasons? The question is how do we basically trust these results? Because as I try to point out that we have done a lot of approximation, we have thrown out many of the interesting complexity that is present in this thing, just to make sure that we can do this simulation in a period that a graduate student can graduate from their PhD and all these things. So one way that how do we basically validate that our simulation result we saw is actually accurate? So initially I showed you that you can validate at a bottom-up approach. You can take one particular peptide, look at the membranes interactions and see whether these matches with the all atom models. But this final snapshot that we showed you, where you saw many weird results, essentially one can say these are artifacts of coarse grain model, that you can see stretched out peptides, you see waters getting inside. Maybe these things are all the way you have mapped the coarse grain model. So what we did is that, why don't we reverse map our coarse grain model to a more atomistic model? So this is something that you can do with this particular thing. So you basically effectively map it out, you back map it from a coarse grain to an atomistic representations, and then you basically subject this particular simulation to 1 microsecond. This is definitely going to be very slow, but you can definitely try to see whether this actually remains stable or not. What we found is that in all this, we ran 2 independent trajectories by taking the last snapshot of this 4, reverse mapped it, and then we basically ran it for 1 microsecond in all atom representations. Even though this is very long simulations and very large bilayer, in both the cases we found that this kind of phosphates getting completely smeared or the waters getting inside is pretty common. And more importantly, in all these cases we started finding that the peptide remain extended in all these particular representation, even though you have not added any external bias. It tells you that you can at least you can see that this particular coarse grain model is resilient to a refinement by all atom structures. And finally, we try to see whether this coarse grain model can actually mimic the experimental observation. At the very beginning, I try to point out that if you change the sequence, the result can be completely opposite. So this is exactly what we try to simulate. Now that we know how to model a coarse grain model for a beta peptide, we try to look at the other one, which is called non-amphiphilic peptide, where you scramble these things, which is not supposed to interact with the membrane that much. And this is exactly what we found, that if you try to put a this non-amphiphilic peptide on the interface, you will basically see that in all these cases peptide does not go inside, it just gets stuck to this particular interface. It tells you that this amphiphilicity was key, and that is reflected in our simulations as well. And essentially the phosphate density, if you compare between non-GA and GA, you can see that as you increase the concentration, you will start seeing that peptide getting phosphates are getting completely smeared, but in a non-amphiphilic peptide this is completely remaining sharp. So this tells you that even in a Martini coarse grain model, the sequence selectivity is retained if you basically map it properly for your particular models. And that also tells you that this is exactly why I think in a coarse grain representation also you can see this kind of sequence dependent membrane activity for 2 different sequences. So here I think I'm just going to conclude here. So basically we try to show you today that we can learn from all atom simulations, go to coarse grain model, try to see whether you can capture a longer timescale event of peptide membrane interactions. In this particular case, we showed that if you increase the peptide to lipid ratio in a coarse grain model for a short chain beta peptide, you can start seeing membrane pore formations, and which can lead to water leakage. And we try to even show that this particular eventual representation of coarse grain model, you can now reverse map it to all atom representations, and that shows that this model remains robust even after that. And finally, what we try to show is that one of the way these short chain peptides can actually get extended is because this has one particular advantage, that it can actually interact with the waters which is present in the other leaflets, and also the phosphate, which will stabilize this transmembrane pore. And finally, we showed that this coarse grain model can also show that this membrane disruption is sequence dependent. So this is essentially, I think, where I'm going to end here. But the thing is that even though we tried initiating this particular project with me, but essentially most of the things actually was initiated when I was doing my PhD. But this is a problem that I could not solve when I was doing my PhD, and I know why. Actually, 2 reasons: because I did not know how to whether our force fields are correct or not. That is one thing. And second is that I did not really have well, maybe that was a problem because I was doing it myself, but now I actually have a good student who actually could do it properly. So Joy Krishna actually mostly did this entire project. He is supposed to defend tomorrow, and Dhiraj also helped. Nomita was the person who realized that in a Leonard Jones Martini water model, this will not work out. If you start doing it in a coarse grain electrostatic water model, maybe things will work out. Pushpita helped out a lot during the analysis of this course. And most importantly, I think I would like to thank Sam, who actually kind of motivated me to think about this kind of directions when I was doing my PhD. And most of the idea or many critical questions that we had at the very beginning during the PhD, which nullified many other directions, came from my PhD advisor Arun Nathiraj and my other mentor Chang Shui during the things. The entire project was done with my institute computing facility, even though I can tell you that these are relatively simplified system, but if you really think about longer simulation, even a coarse grain model, that can also take time. So the funding was basically from our Intramural Fund and ACRB Early Career Research and Co-Research Grant. I also had a Ramanujan Fellowship, which also helped out to buy some of the computers. So finally, as I'll just end with last one slide. So recently we extended this kind of approach of coarse grain model to even looking at bacterial chromosomes, where we try to integrate the models with experimental data called high-C data, which is basically like a genome sequencing data, and we could come up with a segregation of different domains in our models, which was mostly even done by this one of my graduate student, Abdul Wasim. So if you are interested, you can also look at this particular work. Definitely we'll talk about it if you are interested in future. So with this, I would like to thank all of you for your for your attentions, and if you have any questions, I'll be happy to answer.
Yeah, thanks Jagannath for a fascinating presentation, very interesting. And we are now open for discussion. Please raise your hand, virtual hand, if you have a question. Meanwhile, there are a few questions in the chat box. Maybe I'll read it out for you, Jagannath.
Yes, please.
So Abhinandan Das has a few questions. I'll just read all of them together and then you can take it. And some of them you might have already discussed, but let's see. Does water play a major role in allowing the peptide traverse the hydrophobic core of the membrane? Did you try the simulation without water? Antimicrobial activity depends on length or nature of amino acids of the sequence. When do we say an antimicrobial peptide a biomimetic peptide? So lots of questions. And
I think all these questions are coming from Abhinandan. So the answer to this is that I hope Abhinandan saw that this water actually plays a very important role, and especially in the modeling purpose. So the electrostatics of water was crucial here to make sure that this can interact with the peptide that you have, which are having lots of charge residue in the form of beta lysines. So that was the case, because as we show in our control simulation, actually I should not say control simulation, that was the first simulation we did, where we did not have a polarizable Martini water. We found that there is no way that peptide can get inside the membrane. It basically just remained partition in the interface. And as Abhinandan tried to point out, we did not try the simulation without water, because that is not what you should do with Martini. Martini also has a version called dry Martini. So in that dry Martini, even though they call it dry Martini, but it's a very, very humorous name, because Marink is a humorous person. But the thing is that dry Martini only works with membrane, not with protein and all these things. So we did not try dry Martini. And as I tried to point out that definitely the sequence is important. And as you saw from my seminar, that if you even change the amplitude of the peptides, it will matter. It will change the way it will interact with the membrane. It will also affect how you will see the pore formations or not. And last question is that this is called biomimetic peptide, in that sense that this name is popular among the bioorganic chemists, because they always want to mimic a biology or nature. And alpha amino acids is basically a biological peptide, if you think about it. So whenever you try to change the the chemistry of a normal natural peptide, then you basically are tinkering with the peptide, but you are trying to mimicking similar kind of features. So that is the biomimetic peptide. So in this case, beta peptide was having an extra carbon atom in the backbone. So that is completely a synthetic peptide. You will not have any natural counterpart of this thing. So this is exactly why it is a biomimetic peptide.
Okay, thank you. So maybe Dr. Manoj Mondal, please go ahead. Manoj.
Yeah, can you hear me?
Yes.
So I have few questions.
Yes.
First of all, it was very, very, very nice talk. So it shows that the peptides are interacting in a head-to-tail fashion and inside the bilayer. Yes.
That is true. True.
So then, what do you think if you have a single peptide kind of dimer?
Single peptide, only a dimer?
Yes. So dimer, kind of dimer peptide. So you have monomer, this one. If you took, if you take just dimer instead of monomer, what happens? It will go.
No, that is that thing that I try to point out, correct? So if you have only single peptide, okay, it will remain, it will be on the interface, correct?
Okay.
The moment you will start adding the concentration of the peptide beyond a threshold concentration, we start seeing that the tilt angle of the peptide changes.
Okay.
So you need to reach a critical concentration. So this is exactly what people talk about in literature cooperative effect. So this peptide at a very low concentration will have a particular, let us say not head to tail orientations. It will have more like a, surface orientations. And the moment you start increasing the concentration, you start seeing this kind of, head to tail orientations.
Yes. So even if you have a larger peptide, like kind of dimer length, yes. Hello.
I think there is some network issue on Jagannath's side. Let's wait for a minute. Maybe I'll try to just call him and check. Yeah, so Manoj, you can complete your last part of your question.
Yes, so you says the peptide is getting extended when it is in the bilayer, yes?
Correct.
So it is good for antibacterial like properties of peptide, or it's bad? What I am thinking, when its peptide is extended, the pore size, inside pore size is also decreasing, yes? And the permeability, maybe the water may like sometime get stuck in the middle of the peptide, or this is not the case?
So I think the point that it might get, the extension is be necessary for this peptide to get, make a pore is because, this is so short, it will not be able to span the membrane on its equilibrium length.
Yes.
Okay, and basically what I could sense is that, you have a lot of phosphates on both the leaflets, and also have water interface. So if you look at the two terminus of the peptide, one is having a tyrosine, okay, and the other terminus having the last one is the lysine. Okay,
yes.
so they will definitely try to interact with those two interface. And that might be the reasons why you will see that this extension is still possible.
Yes. So what I'm trying to say, is there any relation? So did you find anything like when it is stretched, pore size decrease or something?
No, I think we did not see any pore without any, at a low concentration. And I can tell you only the circumstantial evidence.
Okay.
I will say that at a lower concentration, maybe the pore never formed. Lower concentration peptide is only because it could not actually, it could not really make the tilt, and it could not most likely also got extended. And that is exactly what actually you need at a higher concentration. Maybe they can still make the, extension, because they can still, interact with each other. Okay, with a higher concentration, because these peptides are known to aggregate, also self-aggregate with respect to each other. And maybe you have sufficiently higher concentration to basically aggregate and also interact with the water. But honestly speaking, I really do not know, whether this is good for the, peptides from the perspective of the peptides or not.
Okay. Thank you. Yeah. Thank you very much.
Okay.
Yeah.
Thanks. So next, Ipshita, please go ahead.
Hello.
Hi.
Yeah. So I just want to know that, like, you notice the aggregation leads to the vertical orientation of the peptides, right?
Aggregation leads to vertical. That is basically the thing that we try to see. Actually, at higher concentration only, I see that vertical orientation happens. Yes.
That's why actually the hydrophobic mismatch comes. And like, I just want to know, did you see any bilayer thinning along with the peptide stretching?
So basically, I think in this case, what I showed you is that by bilayer thinning, you most likely means that whether bilayer get expanded or not. Correct?
The hydrophobic thickness is
So
read.
basically, as I showed you, the bilayer thickness profile I tried to show you, correct? In the plot, maybe I can.
Yeah, there are some patches.
I can show you one more time if you are. I can show you one more time, and maybe let us see if I represent it.
No, I remember. Yeah, it was that. Just
Yeah.
no, I just want to know that. Did you find any correlation between the aggregation and the vertical orientation? Because for the single peptide, lower peptide to lipid ratio, there is no. The peptide is not the tilt angle is not getting changed.
Correct. So definitely, I
Yeah,
think there is a correlation between the aggregation and the tilt angle. Now I am saying
yeah.
that this is a correlation. I don't know why it is changing the angles.
Okay, okay.
Exactly why I don't know exactly why it is changing, but you are right that at a very low concentration you don't see any change in the angle.
Change.
But
Yes.
once you basically start seeing these things at a higher concentration, it starts appearing. Essentially, if you look at some of the literatures by someone called Hui Huang, he had done extensive experiments on many of these antimicrobial peptides, and the way he measures is he quantifies this thing. He uses something called orientational circular dichroism, orientational CT. So in that particular thing, they showed that also in an experimental way that the orientational CT actually changes with increasing peptide to lipid ratio. Okay,
Okay.
and their hypothesis based on their experimental data is that it is to do with the change on the orientations
Okay, and one more
at a
thing
higher concentrations.
what I just want to know that I saw in the all atom simulation for
Mhm.
higher PL ratio, that is probably 7:128. Just
Correct.
you just extend the water shell. So is there any specific reason not?
So basically, otherwise, what you will see is that if you have because you have many of them, correct, on the surface. So what you will see is that if you don't increase the water cell, so many
No,
of the peptides will go on the other interface
no,
actually.
no,
So
no.
it will like the periodic boundary condition will have reached
No,
actually.
can you just extend the bilayer across the XY plane? I just want to know that if it can change the result, like instead of the peptide aggregation, one may,
I get that what you are trying to point out,
yeah,
whether you can increase the bilayer thickness. So bilayer span actually, I know bilayer patch size.
yeah,
Let us say
XY
120
plane.
to 336 or something. So the point that we basically found is that we thought that we want to increase the peptide to lipid ratio. So then I have to increase the number of peptides as well. Correct. If I want to make the similar concentration at a larger
No,
bilayer
no, no,
space.
no. I'm not talking about the bilayer thickness. I'm just saying that because you extend the water shell, so
Correct. Correct.
you have to put the peptides far from the bilayer surface.
That's
But
true.
from your 1:128 PL ratio simulation, you already noticed that because of the amphiphilic nature of the peptide can penetrate to inside beyond the phosphate level, right?
Correct.
So if you expand the bilayer in XY plane, if you just increase the dimension in the XY
I get
direction,
it. Yeah.
yeah. So
So you say that you will have more peptides to accommodate on XY plane, correct?
XY plane, huh?
That might be a good idea, but I did not try it because I thought that that will increase the total number of particle size, and maybe that should have been tried actually.
Okay. Thank. That's all.
No, that I understand what you're trying to point.
Yeah.
That would have been a better idea. You were right. Now, if I think about it back now, so maybe that would have been a good idea. But I did not try it because I thought that it is taking quite a lot of time. So why don't I just increase the number of waters, which is maybe it
Oh,
will not add that much actually.
okay.
Okay, next Krishnendu.
So, sir, can you hear me?
Yes.
Okay, so when you build the Markov state model, you considered two states. One is solvated and the other one is adsorbed state, right?
Correct.
So how do you distinguish these two states?
So this
Means,
is based on the minimum distance.
okay,
Minimum
so
distance from the peptide to the what shall I say, like the minimum distance of the peptide to the lipid, lower liplet or upper liplet. Sorry, upper liplet. Yeah.
okay, so when we usually build Markov state model, there is a step for dimensionality reduction where we choose a feature for
Correct.
dimensionality
Yeah.
reduction.
In
So
this case, it was simplified. There was only one dimension. Correct. Only the minimum distance between the peptide and the lower upper liplet of the lipid. So you do not need to reduce the dimensions any further, because you are only dealing with only one dimensions. You
Okay.
just cluster your data on that.
Okay, so you have performed k-means clustering, right?
Correct.
You consider 500 states. So
Correct.
why not just if we are clustering based on the minimum distance? So if minimum distance is below some cutoff, it is a solvent adsorbed state. Otherwise, it is solvated state, right?
Correct. Correct.
So you can only consider two states. What's the point of considering 500 states?
That's because I wanted to make sure that I can build a basic Markov state model with in the microstate first, because I have to discretize the trajectory, correct, first.
Yes.
So
So
that is
you can
why.
just discretize between zero state and one state, right?
So
Considering just
basically, what I did is that after we build the MSM on, let us say, 500 discrete cluster centers, then
yes,
I basically once I have the Markov state model, then I basically
use PCCP plus or
because
something like that.
even here we did not even did PCCA or
Oh,
anything. All we did is that we tried to say that which microstates, let us say you put a distance criteria, which microstate belong to this cutoff, which will be solvated, which microstate belong to this cutoff, which is basically rest of them, which is basically the, what shall I say, the adsorb state. Okay, and then for each of the microstates, since you already have done a microstate model, you know the stationary population of each of the microstate, correct? You just sum them up, then pretty much you will get the corresponding macrostate population.
Okay. Thank you.
I'll just ask something quickly, follow-up as a follow-up question. So when
Yes,
you are
yes.
building this Markov state model,
Yeah.
so do you see enough unless you see enough number of detachment events, will your transition matrix be correct? So I have never done it myself, but
So
I always find it curious that when you have really rare events, that reverse process, how do you build
yeah.
the matrix?
So in this case, actually, what we do in all the times in Markov state model, you enforce the reversibility.
Okay. How do you do that?
How do you do it? Basically, you just do the, what shall I say, in any Markov state model, what you do is that you let us say you calculate how many counts you will see on the front. Okay, if you go by this, let us say transition from A to B, you basically make sure that from B to A also you will see the similar kind of transitions, and you sum them up and divide by 2.
So just average.
Just average. That is basically called, you enforce the detail balance by enforcing the microscopic reversibility. That is not accurate, but that is how people generally do it.
Yes.
You can also try to build an irreversible microstate model also, like only by one way, and then
has it been benchmarked that if you have a really large difference between K on and K off, does it really reproduce the numbers?
99% cases you will see that your off rate is very, very off from experimental data. Two reasons: experimental off rate also have very large uncertainty, but you are also not having that many events going towards the other direction, correct? Because you don't see it. So essentially, unless you basically enforce it, you will not be able to even calculate a Markov state model for the other events.
Right.
So nobody has even benchmark. One way to do will be that you can try to do a simple, maybe it will be interesting to look at, like one can do a very simple two-state model where you see that both sides happening, and then see without the reversibility and with reversibility what actually happens.
So is it possible to build Markov state model with some enhanced sampling like replica exchange molecular dynamics?
Actually the problem is that in Markov state model the idea is that you are looking at transition, correct, between one particular frame to another particular frame. So if you have you need to have a time unit, correct?
Okay.
Each of the frame, for the next frame it has to have a time correlation, correct? So unless you have a way that you can reweight your enhanced sampling based trajectory in somehow, then you will not be able to use them for a Markov state model. People used to do that sometime in metadynamics when they do it in an infrequent way, like Protus does actually. But I think that is not accurate actually. Like if you really want to have Markov state model, you can do a normal k-means clustering for any data. But if you are looking for a kinetic information, then you need to have a kinetic information preserved in that simulation also. So none of these replica exchange or enhanced sampling will be able to do this properly, but replica exchange will be useful because you can try to do adaptive sampling from those replica exchange trajectory and try to enhance short trajectories and try to increase the number of population in your state.
Okay, so maybe the last question, which in the chat box, Shutonu Mukhopadhyay, can we use cyclic peptides instead of beta peptides?
Yeah, there is. So basically, most of the things I learned from Durba's paper, Durba Sengupta's paper. Durba has already done this actually. So in her way back in 2011-12 paper, you will see a biophysical journal paper or a BBM membrane where they have used cyclic peptide, which is synthetic peptide, and that also can show pore formation in a Martini presentations.
Okay, it's another one. So maybe this is the last one. Shoshti, go ahead. Yeah. Shoshti, go ahead. Yeah.
Hello, sir. Can you hear me?
Yes.
Okay, so my question is, instead of doing long simulation of 2 microsecond we have done, so can I use steered MD simulation to pull the peptide inside the membrane?
Yeah, people do that actually. Many times you will try to look at pore formation by these things, where you can try to do steered MD and then
it can save time.
it can save time, but only problem that you will see two problem is that is non-equilibrium. So
Yes.
you should look at, there is a paper which is the most likely most popular, paper on antimicrobial peptide. Is called Antimicrobial Peptide in Action by Maring, 2006 paper in JACS. Okay, and there they showed that if you use steered molecular dynamics based simulation to generate pore, okay, that pore will not be stable actually.
Okay.
But in that paper, there is a issue. They have not used counter ions when they have done the simulation. So they saw pore formation very fast, which is unusual actually. In 200 nanoseconds, they saw in non-atom simulations pore formations. So
also in this case, I think you have another aggregation coordinate. So it is not just the vertical movement,
correct.
but
So
there
you
is
have to also
another
think about
self-assembly. That self-assembly
correct.
coordinate becomes important.
Important because in a steered MD, you do not know then which particular peptide you should pull inside, correct? Because you have an aggregation
Yes,
also
the
happening in the
direction
same
is
time.
important. Yes.
So that will be tricky.
Yeah. Okay. I had several questions, but I think we should stop at this point.
Okay.
Because there is also, you have this other talk to attend.
Yes.
It's almost 5:30.
Yeah.
So whatever I had, I'll discuss with you separately.
Sure, no problem.
So again, thanks a lot for coming here virtually, and I think students have benefited a lot from your discussion.
Thank you, Suman.
And I extend this open invitations that next time when things are normal, you should visit us.
Of course, I will come.
Sure.
Yeah, and maybe I'll see you tomorrow.
Yeah.
Yeah.
Sure.
Okay then.
Yeah. Take care.
Bye.
Bye, everybody.
Bye.